feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. And I'm right where I belong in my happy place. And I'm right where I belong in my happy place.
Hi, and welcome to the National University of Singapore Yong Lu Lin School of Medicine's Healthy Longevity webinar. Thanks a lot for joining us tonight. I'm joining you from Shanghai, and we have our speaker, Professor Riccardo Marioni, who will be with us tonight from Edinburgh, I think. That's what the background says anyway. I can tell because it's cloudy and there's no sun. I'll be coming back to him in a minute. I want to remind everyone to use the Q&A function to ask questions tonight. But before we get to Riccardo, I'm going to bring on Joshua Rain, who's a research fellow at the NUHS Center for Healthy Longevity, and he'll talk about evaluating the transmission risk of amyloid beta peptide via ingestion.
Thanks very much. Hi, as mentioned, I'm Dr. Joshua Rain of the Mechanisms Underlying Behavior Lab, and today I'll be discussing one of the key players in Alzheimer's disease, amyloid beta, and its transmission risk via ingestion. Now, that might sound a little scary, but don't panic. There is no cause for alarm as of now. Alzheimer's disease is a neurodegenerative condition that typically manifests in people over 60 years of age, resulting in progressively worsening dementia and cognitive ability. The full causes of the disease are complex, and differentiating symptom from cause is challenging. Much focus has been placed on amyloid plaques and tau tangles, and it is against these targets that current antibody treatments are being developed. Alarmingly, it was discovered in the last decade that Alzheimer's pathologies can be transmitted through medical procedures. People given contaminated growth hormone as children exhibited A-beta pathologies typical of early-onset Alzheimer's disease in just their 40s and 50s, while possessing none of the genetic risk markers. The mechanism of this transmission has been attributed to the prion-like properties of A-beta. Prions are small pathogenic misfolded proteins capable of self-replicating by causing the misfolding of native proteins. Such self-replication is cited to allow A-beta to act as a pathogen, propagating distinct strains with morphology and composition of the plaques based on their origin. The most well-documented example of prion transmission was mad cow disease, a health crisis caused by the consumption of meat containing prion proteins, resulting in sudden onset Creutzfeldt-Jakob disease in humans. The key factor in the transmission of mad cow disease was the ability of prions to resist digestion, a characteristic that is yet untested for A-beta. If the potential for amyloid beta to act in this manner exists, it would lead to an upheaval of worldwide food safety practices. Fortunately, we had the ideal tool available to test this cross-species transmission risk. A study from our lab generated an A-beta fusion protein that aggregates under certain wavelengths of light. When expressed in zebrafish neurons, this revealed that the A-beta peptides and aggregates formed caused physical damage and even resulted in some neuron bursting, as shown in the video. This fusion protein was also expressed in some fly larvae to further study the properties of its aggregation, but this expression in fly larvae allowed us to use them as feed for zebrafish, which they readily consumed. This let us test the transmission of A-beta aggregates to wild-type zebrafish by ingestion. However, we did expect this transmission to be minuscule, so we needed sensitive methods to test this possibility. We can easily detect amyloid plaques in the developing fly larvae pre-feeding in the images above, but we don't observe any plaques in the controls below. From here, the experimental feeding was split into two paths. Firstly, a short-term feeding used to test for gut retention. These zebrafish were fed 20 larvae each over 24 hours, and then allowed another 24 hours for clearance of gut content. These guts were extracted and screened for A-beta by the methods listed. The second path was a long-term feeding, testing for seeding and accumulation in the brain tissue. If any undetected amyloid seeds were surviving digestion and being transmitted to the brain, they could potentially seed new plaques. These fish were fed hundreds of transgenic larvae per week for 3 months, and the brains were analyzed in the same manner as the guts. Fortunately for all of us, there was no evidence of any A-beta transmission, neither retention in the gut tissue nor accumulation in the brain. Here's a small selection of our mass spectrometry, immunoblotting, and histology data, where the green indicates detection in the feed, while red shows no transmission to the zebrafish. There are a few additional factors to consider, however. Firstly, the transmission may simply take longer to manifest than we observed. The individuals given contaminated hormone treatments we discussed earlier took decades to show their pathologies. Secondly, different A-beta strains have variable digestive resistance, which could increase the risk of transmission. And finally, fish A-beta is only around 70% similar to human. Hence, the absence of A-beta seeding in our experiments may be due to this sequence dissimilarity, rather than the inability of A-beta to act as a prion seed via ingestion. Livestock animals, however, all have an identical A-beta sequence to humans, increasing the risk of interspecific seeding. We could test this possibility further by generating a knock-in zebrafish line, expressing human A-beta, and testing again over several years. But to conclude, as of now, there seems to be no transmission and no cause for alarm. I'd like to briefly thank the contributions of Associate Professors Jan Gruber and Nick Tolwinski of Yale NUS and the Singapore Ministry for Health for making this research possible. Thank you very much for listening.
Well, it's a very interesting study with, I think, the outcome we were hoping for. I want to introduce Dr. Riccardo Marioni. He's a professor of molecular epidemiology and aging at the Institute of Genetics and Cancer at the University of Edinburgh. He has a background in epidemiology and biostatistics and is a co-PI on the Generation Scotland cohort. His team integrates multi-omics data with electronic health records to better understand the biologic mechanisms that underlie disease outcomes and facilitate improved risk prediction. So tonight he's going to be talking about the topic DNA methylation-based predictors of lifestyle and disease. Thank you for joining us, Riccardo.
Thank you so much, Brian. Yes, it's a pleasure to join you today, and I'm looking forward to the active Q&A session afterwards as well. So I will just share my screen now. So yes, thank you so much. As Brian mentioned, I'm going to discuss with you today some of our ongoing work on DNA methylation and its associations with lifestyle and disease. So some of this work is available in preprint, and some of it is currently unpublished, but hopefully in preprint quite soon. So as a very general introduction, when we think of DNA in fairly lay terms, I picture this as a very long ladder with many billion rungs. And this is the same pattern, the same rungs in every cell in our body. We're 99-point-something% identical across all of these rungs, but where we differ, that variation helps explain differences in our phenotypes, so things like eye color, height, and so on, and to a certain extent predisposition to disease outcomes. So this ladder and these rungs stay fixed. However, there are potential chemical modifications to DNA. So this is the study of epigenetics, where we can look at these chemicals that can be added or removed from DNA over time. So this is a dynamic process, different in every cell in our body. And there are certain points along the DNA ladder where these groups can be added. And we can see these, or we can currently measure these in around 800,000 positions, 800,000 rungs on the ladder. using Illumina technology, and we've done this at large scale in some of our cohort studies. Now, very crudely, we can think of these modifications acting a little bit like dimmer switches. So it's kind of turning genes up or down, which results in more or less protein being produced, which in turn will relate to health and disease outcomes, which we can measure either in the clinic or over time through things like electronic health records. So in the cartoon here, we have 4 different cells, and at one of the rungs on the DNA ladder, we have, in 3 of the cells, we have a methyl group attached to the cytosine, so we call this CpG methylation, and at the 4th, we don't have a methyl group attached. So we say this person has 75% DNA methylation levels at this particular position in the genome. So all of the examples I'm going to discuss with you today use data from the Generation Scotland study. So this is a longitudinal cohort that began in 2006, with over 20,000 people from around Scotland being recruited to the cohort. There was regional sampling and also family-based sampling. So we have up to 4 generations per family. The age range at the start of the study was between 18 and 99. And interestingly, we also have permission to recontact all of these volunteers. So if we want to do a recall by genotype or a recall by phenotype study, that's also possible. The volunteers additionally gave us permission to access their medical records, which stretch back good quality data anyway, back to around 1980 for primary and secondary care records. And presently, from the individuals that provided biosamples at the start of the study, we've measured DNA methylation using Illumina technology in 19,000 of the participants. So where to start with methylation and analyses? I think aging is the most obvious example. And by aging, I mean chronological aging. So here, if we think back to the cartoon I showed earlier, we see the individual with 75% methylation at a particular CpG site, so a particular rung on the DNA ladder. And from the plot, we see that that individual's chronological age is somewhere in the region of 30, but based on the methylation levels at this CpG site, relative to the average population member, they probably resemble more an 80-year-old than a 25 to 30-year-old. So this this was first explored around 10, 12 years ago and led to the concept of biological aging and age acceleration. So why does this individual have methylation levels that might be indicative of the levels of an older individual? Is it down to, it could be something as trivial as a sample mix-up or a mislabeling, but it could also be due to differences in health behavior and disease. But instead of exploring this at just one CpG site, we can look across all 800,000 sites and then try and narrow that down to get a signature that is predictive of age in our cohort. So the method we typically use for this is something called penalized regression. So we start with all 800,000 sites. We use this kind of machine learning approach to identify a subset of those CpGs and the weights for those that give us the best prediction of the outcome. So in our example, we have here chronological age. This is a study that was published last year in Genome Medicine by Elena Bernabeu from the group. And here we use data from Generation Scotland, in addition to other publicly available data, so 24,000 individuals in total. And we asked whether linear effects, such as we can see in the CpG on the left-hand side, and nonlinear effects, so the quadratic association, or we modeled it as quadratic associations just by squaring the CpGs, or the values for the CpGs, whether that's predictive of chronological age. And what we saw with this epigenetic clock in a test set is that we're able to get a very strong correlation with chronological age to the extent where the median error is 1.7 years. So this means that for 50% of the test population, we're able to predict their chronological age to within 1.7 years. So a really accurate prediction of chronological age. And then we can ask whether deviations from that are associated with biological aging outcomes, so things like disease, longevity, and so on. However, one complication here is how many CpGs to start with. So we're in the realm of extremely high data here with 800,000 columns and 20,000 rows. And this does result in some limitations to the machine learning approaches we take. For example, it's far too high dimensional for methods such as random forest to work. And even with elastic net or penalized regression, this can still result in overfitting to the cohort that we're training on and resulting in predictors that don't generalize as well to external data sets. So a current preprint from the group, this is work by Yipeng Cheng, a PhD student. This is looking at methylation in relation to type 2 diabetes and exploring the effects of feature pre-selection prior to training our model. So on the right-hand side of the slide here, we have our 800,000 CpGs. One simple approach we can take to reducing the size of the data set is to run principal component analysis, PCA, on the data and take the first however many PCs that explain a good proportion of the variance in our data set. Secondly, we can also, a lot of the CpGs on the array are relatively invariant, so not much difference between individuals. So we can filter those out and then ask if we just take the most variable CpGs on the array, can we use those for the prediction models as input features? And then finally, we can also ask if epigenome-wide association studies, so analogous to genome-wide association studies, have been run for our trait of interest, can we use the literature and the previous findings to identify a subset of CpGs that might be good for training? This approach is ideal, but unfortunately, for a lot of traits, there's not going to be EWAS literature out there, or at least it'll be relatively low power within the studies. So the approach that Yipeng developed is one that we've called related trait-based feature screening, RTFS. And here we take commonly measured traits that are known to correlate with health and lifestyle. So things like BMI, alcohol consumption, smoking, cholesterol, and so on. We first run our penalized regression models to identify CpGs associated with each of these traits. And we then take the union of those CpGs to create a fairly parsimonious set of features that we can input into our elastic net predictor. And in Yipeng's study, we compared the performance of the RTFS subset with the other methods that I described, training in Generation Scotland, testing in a subset of Generation Scotland, and also testing in a subset of the CORA cohort, a German epidemiological study. And this was for incident prediction of 10-year onset of type 2 diabetes. And what we saw was a very similar performance of the RTFS predictor with the predictor that used the CpGs identified from the existing studies of incident type 2 diabetes. But both predictors, although not shown on the plot here, did outperform the other methods we considered, so PCA or the variance thresholding. In addition to disease outcomes, we've also recently explored associations between methylation and lifestyle factors, including traits such as cigarette smoking. So this is fairly established in the literature that we know that there are individual CpG sites and combinations of CpGs that are highly correlated with smoking status. So we refined that predictor slightly in this analysis where, again, using the CpGs that have been identified in the literature as a priori to be associated with smoking, we subset to those, we trained a predictor, we tested it in another Scottish cohort called the Lothian birth cohort of 1936. And we see really good discrimination between current smokers, former smokers, and also with never smokers. Interestingly, in the Lothian cohorts, we also have access to, in addition to the blood methylation from various time points, we have post-mortem brain methylation as well on a subset of the cohort. And what we see here is really rather interesting. So the top panel there, we see a specific CPG that was measured in the blood of the individuals, and we see that it discriminates smoking status quite clearly across the three groups. On the bottom panel, however, we see very little association with smoking status at that particular CPG. We then compared these results to those from different tissues, so here the hippocampus, and we see that whilst the top CPG does a very good job of discriminating smoking status in blood, it pretty much flatlines in the hippocampal tissue, whereas on the bottom, the CPG that's invariant in blood or is comparable across the groups actually shows very strong discrimination in the hippocampus. So it may well be that smoking and lifestyle factors influence the methylation patterns in different tissues differently. So are there different genes or pathways that might be affected within the brain compared to blood with smoking? And that could be really important in helping us identify the mechanisms through which smoking relates to things like cortical thinning, which is established in the literature. So moving on from smoking to alcohol consumption, this is a preprint, I think it came online last week, again by Elena, and similar to the previous studies, again, we filter our methylation to around 4,000 CPGs that were identified from the literature. We use an elastic net predictor in Generation Scotland. We compared this for males and females together, males and females separately, and we found that the best predictor was just when we include everyone. Similarly, the optimal predictor also used data regardless of whether the self-reported units of alcohol consumed in the last week was reflective of the normal week or was more or less than individuals usually drank. And I think that's quite important because even if one's being very honest, it's still difficult to accurately note how much alcohol you've consumed in the last week. So, for example, if you have a glass of wine with dinner, are you aware how big the glass is? Or can you remember how strong the wine was? So things like that, that even if you say I have one glass of wine, that could be quite a different unit of ABV profile per glass. So what Elena saw was that for this alcohol predictor, it correlated very strong, well, moderate to strongly in 4 external cohorts of differing ages and ancestries. So correlations between 0.4 and 0.5. Most interestingly there, I think, is the sister study, which is a cohort of white, African, Hispanic ancestry women from the US. So unlike polygenic scores, which don't tend to translate terribly well across ancestries, methylation scores do seem to translate quite well. We see in the bottom left of the plots that a decent ability to discriminate heavy drinkers from non-heavy drinkers based on recommended thresholds. And on the right-hand plot, something which I think is really quite interesting and maybe alludes to the fact that self-report is not the best metric for things like, or the limitations of self-report for factors such as alcohol and smoking. We looked at both the methylation predictor and self-reported smoking in relation to 4 measures of global brain health. And we see no associations between self-reported alcohol and the brain health, but we do see associations of greater magnitude and also statistical significance for the EPI score. So this is with increased white matter hyperintensities, smaller total brain volume, less grey matter volume, and so on. So in the final example here, we have, again, a fairly similar pattern to the previous analyses, this time looking at methylation and metabolic traits. This is work by a PhD student, Hannah Smith, and again, a preprint should be online in the next few weeks. So taking data from Generation Scotland, this time for 6 different metrics of metabolic health, so BMI, waist-hip ratio, body fat, and then 3 blood biomarkers from Generation Scotland. We ran epigenome-wide association studies to identify individual CPGs that might be associated with these traits. I'm not going to focus on that today. What I will focus on, though, is the training of the epigenetic scores of the trades. And we projected those into the Lothian birth cohort of 1936 and also into the Helios cohort, which is based in Singapore. So what we found, again, reflecting the results from the sister study for the alcohol analysis, quite a lot going on in this slide, but in essence, our methylation predictors of metabolic health do a really good job in both a Scottish test set and a Singaporean test set. So here in the darker of the greens, we have the variance explained over and above age and sex for Helios. In the lighter green to the right of it, we have the results from Helios for the Chinese ancestry participants. In the blue, we have it for the Indian ancestry, and in the purple, we have it for the Malay ancestry Singaporeans. We're still digging into some of these findings. So, for example, I think an interesting one is for body fat percentage, where for the Chinese and Malay ancestry individuals in Singapore, we see around 10% of the variance being explained, whilst in the Indian ancestry subpopulation, we only see around 3% of the variance in body fat being explained by the EPI score. So that's pretty much the end of the talk. So to summarize, just like to emphasize that methylation patterns are very much correlated with lifestyle behaviors and perhaps give us a truer or certainly the more objective estimate of behaviors. They augment disease prediction, so for things like type 2 diabetes, and they translate well across other populations, so different ancestries, ages, and backgrounds. However, there are numerous challenges with these data analyses. So specifically, in the era of quite intensive methods being available to analyze data, are there ways that we can cleverly shrink down the input size to begin with to give methods, so nonlinear methods such as random forest, Bayesian regression trees, a better chance of improving prediction further, as opposed to just considering linear features or throwing in all possible CPGs for these analyses. So with that, I'd like to thank everyone in the group, past and present, all of the Generation Scotland team, particularly the volunteers, and also to all our collaborators who helped with the analyses presented here. And thank you very much for listening, and very happy to take any questions if there are.
I'm sure I can dredge up a few questions. So let's see where we go. Also, if you have questions in the audience, please submit them. Shiva is coordinating them, and she'll be asking them in a few minutes. We have plenty of time for questions. So now's your chance to ask all things methylation and epidemiology. I'm sure there's no questions that Riccardo can't answer. So I'm really interested in the cohort, particularly, I mean, it's a nice cohort. It's so big and across such a wide age range. But you have family data nested in there too. So was there anything, I haven't seen family data on methylation as it relates to aging or disease in the context of such a big cohort. Is there any particular insights that might come from that?
That's a very good question. So some colleagues in Edinburgh have actually looked at parent of origin effects on methylation. I forget where that study was published, but we've not focused too much on the family structure other than to ensure that when we are training or running training predictors or looking for CPG correlates, that we do account for the relatedness. It's the sort of thing that I think with longitudinal data and also, as you say, the family structure would be interesting to see whether certain risk scores are nested within families. And by using the linkage to the health records, we can ask if there's, say, siblings, one of whom goes on to develop a disease that was also present in the parent. Are there any signs from their biomarkers or methylation, years in advance of that diagnosis?
I suppose if you could you have power to do this, you could ask, when you have a set of methylation sites that predict something, you could then ask in a family, are there subsets of those methylation sites that drive both the prediction and potentially some outcome?
Yeah, that's an excellent question, and it's something I'm not entirely sure why, but we're yet to explore. I think there's so many possible ways we can look at things, but certainly the within family and between family effects, it would be great to look at. I think our biggest pedigree is around about 30 individuals, so there's quite a deep structure within the cohort, and as I mentioned, up to 4 generations per family, typically 3 generations.
That's interesting. I was going to ask a question about specific sites anyway, and then I ask it in the context of families, but let me take it back from a broader perspective. One of the challenges, and I'm thinking about methylation clocks for aging here, but it could be for any outcome. One of the challenges, I think, for these clocks is they're markers, yes, but can they tell us anything functionally? So can you work backwards from methylation sites to gene expression and particularly even functional pathways in cells that might underlie the predictive score? And one way I know Morgan Levine's tried to do that is to identify, say, 300 methylation sites that predicts aging in her context or mortality, and then try to figure out what those methylation sites are linked to and how they fit into hallmarks of aging. But it also struck me that, if for any individual this is a hypothesis, I don't know if it's true, for any individual that's particularly elevated age or reduced age, and again, I'm using aging as an endpoint here, but it could be anything. Can you find clusters of sites, do you think, that are out of the normal range that are driving that prediction in each individual, and look at the relationship between the specific sites that drive the person in one direction or another? That may be a better way at pointing to pathways than just trying to link methylation sites to pathway.
Yeah, a lot to unpack there.
Sorry.
So I think there's so as I alluded to a little bit with the smoking example, one thing that would be nice to explore in more detail in terms of the what pathways are associated with or what genes pathways are being identified is to look across different tissues, because as you say, within blood or whatever tissue you're looking at, is it a biomarker or is there something more causal or potentially causal happening? One way that you can explore the causality is through, I suppose, approaches like Mendelian randomization, where you can ask if there's is it going from a gene or from genetic variation to change in methylation to change in gene expression or change in trait, or whether it's are you going from, is it changes in methylation that drive changes in the trait, or I think more likely changes in the trait drive changes in methylation. So smoking and alcohol being obvious examples where that would almost have to be the case. I think with the aging patterns as well, it would be interesting to look at, can we talk about the clustering, or if there are outliers potentially as well? So do people accumulate different, are they at the extremes for certain CPGs across across the genome? And also whether the age signatures we observe, again, across tissue, we're just a way to, or we're collecting saliva samples from at the moment, we have another 10,000 people recruited to Generation Scotland. So to ask, do those patterns match up with the patterns from blood? And I think hopefully by comparing across tissues and that might give us a better indication as to which sites are reliably associated with, or at least pan-tissue associated with the traits. And does that kind of point us in certain directions in terms of pathways and mechanisms that we can be more, or we can prioritize for downstream, whether it's.
I like the idea. I like the idea of choosing epigenetic sites that from EWAS are associated with a particular trait. Maybe that's a way to give yourself a better chance of getting back to some functional meaning for what's going on.
Yeah, well, I think one of the challenges there is that a lot of the EWAS literature, it is looking at linear associations between CPGs and outcomes. So it's I guess where with the diabetes analysis as well, it's a good trait to look at because there have been quite a few fairly large scale studies. But if we take something brain-based, so dementia, for example, there are relatively few studies and not many large scale ones. So that's kind of where the thought of going to CPGs that are associated with biomarkers that are more commonly measured across all cohorts. If we take those as the inputs, then that's maybe a better starting point for training predictors of disease.
I was going to ask the question about linearity versus non-linearity. I mean, that's a difficult question for everybody building clocks because, do you fit everything into a linear model because it's easier to work with linear data, or both in the terms of the input being the methylation sites, but also in terms of the output? One of the things that a lot of people do is they assume that the variance is the same across the life course, which strikes me as highly unlikely for biologic age, at least. And so, how do you deal with this in your context?
Yeah, so we looked, so in the paper I mentioned, we considered log age as well as linear age for the training. And you do see these, certainly it's quite clear non-linear associations in early life, and then kind of more of a plateau effect in later life. I think we'll be in a good position to explore the early life stuff better with the new cohort where we're recruiting people from age 12 and above, so we can hopefully try and see what happens during the formative years more than, because I think a lot of the current clocks and studies have been very much biased towards middle-aged adults. So it's trying to see what's changing and how quickly it's changing during teenage years would be really interesting, and also how that relates to the obvious biological changes that are happening during that period as well.
Yeah, we finished. We were talking about this before the show. We haven't published this yet, but we finished an analysis of 10,000 Singaporeans of all three major ethnicities across an age range. And interestingly, that first set of 10,000 in SG10K has a birth cohort in it. And so we tried 4 methylation clocks that were already out there. And those methylation clocks were really not made for any of these ethnicities, although there were some Chinese Han people in some of the clocks that were some of the clock construction data sets. But interestingly, we tried to apply it to the birth cohort, and 2 of the methylation clocks, and we use Zhang, Horvath, PhenoAge, and GrimAge, but I can't remember which clocks are which. But 2 of the clocks had development built into the clock, so they had data from children in it. And 2 of the clocks didn't. And I guess not surprisingly, we found that the clocks that used development in their construction were better able to predict age at birth. And so it's not a surprising result, but I think it reinforces the need to use as wide an age range as possible.
Yeah, I think, absolutely. And I guess there's a bit of a paucity of data sets at the same scale in the younger and the oldest old as well as to something that's going to hamper model development or at least for models that could be generalized across the board.
Yeah. And oldest old also brings in selection bias too for people that are still alive. And so that's also a challenge. It was interested about the brain methylation data. We have a paper submitted now looking at making a biologic aging clock from postmortem samples of brain using lipidomics. We really wanted to see more what we could do with lipidomics, whether it's possible to build a clock or not, and we were able to get data from a lot of postmortem samples. And so in writing that paper, I was kind of struggling, right? Because we use that clock to predict chronologic age of the individual. And but the in this case, the lipids that we identified, or in your case, the methylation sites, are skewed toward functioning of the brain. And so I'm curious about how much it's telling us about the whole body versus the organ where the clock is done. And that's true for any methyl, even serum. I mean, it's one organ, right? So the question is, what is systemic aging and what is organ aging, and what are we measuring when we look at one tissue? And that could be for smoking or anything. Again, my brain is fixated on aging. Sorry about that.
No, that's another excellent question. I think it's I also wonder with brain data whether there's what the effect of the dying process has on any kind of omics readout from brain tissue.
Yeah,
that's another major challenge, I think. So, yeah, and there's not an awful lot anyone can do about. It's not a tissue you can sample during life, so you kind of just have to deal with what we get. And it's amazing that people are willing to donate brains to these studies. But I agree. I think that's the pan tissue thing. So obviously, Horvath's original clock looked at I think it was like over 50 different cell types and tissue types. But it would be I wonder if there's something that could be done around if you look at things, or as we discussed beforehand as well a little bit, what is healthy aging? And if you could identify CPGs associated with healthy aging, let's say, are they also clock CPGs? Do they change with chronological age, which might point towards sites that are not only indicative. So if you've got your whatever CPG with a association with chronological age, and somebody's down here, that's actually and that CPG is linked to healthy aging as well, then maybe there's more evidence towards it being, slowing down the aging process or being causal in aging and health. So I think it's difficult separating out what might be a forensic predictor from what might be a biological one, and if there's different pathways going on in just the passing of time as well as the changes in health.
This is why we've spent a lot of time lately trying to make clocks from lipids or metabolites or proteins, because once we identify the major features that go into the predicting the number, we can go back and we're one step closer to function, because with methylation sites, you're then guessing what they're doing. Whereas if we have a protein or a lipid, we may have a chance of knowing what it's doing. And so we're using those more for trying to work backwards from clocks to function parameters of aging. And so that's sort of why we did that study. But I'm puzzled about what it's really telling us a little bit. The alcohol study, we all know heavy drinking is bad. You showed that very well with can you differentiate between moderate drinkers and abstainers? Are there abstainers in Scotland? And can you differentiate them with moderate drinkers? And what are the risk factors there?
So with that analysis, we were it was looking, so the classification was actually based on the NHS guidelines at the time. So I think it would have been 21 units a week for males, 14 for females. Those were the thresholds that we tried to discriminate. Anything above or below that. In previous studies, there was a meta-analysis with the Charge Consortium. They looked at, I think it was more like alcohol or levels for alcohol use disorders. So, higher threshold for separating folk out. Again, the abstainer point is an interesting one as well. And also, I suppose you've got non-linearities in the CPGs, but you've also got wildly non-normal distribution of the phenotype. It's not something we focused on too much. We kind of just bulldozed through as in a traditional regression framework. I think that's where you can maybe explore calibration in the predictors as well to see if they're doing better at one end of the spectrum or the other end of the spectrum, or if it's, is it just able to separate never drinkers from current or light drinkers? But it did reasonably well across the, so you do see a dose response with the predictor that we've developed. I think it's nice.
Go ahead.
No, I was saying it could be nice for cohorts where, or potentially to impute variables. So if certain phenotypes haven't been measured or are difficult to measure, then perhaps you can use methylation to proxy for those, be that smoking, alcohol, or potentially protein levels as well. We've done a bit of work on that to show that you can get reasonable proxies for over 100 circulating proteins. So it's a way to kind of potentially boost data sets as well.
Well, we have a bunch of questions, so I'm going to bring Shiva in to ask some of those questions. Thanks, Shiva.
Hi, thank you so much for such an interesting presentation. I actually have a question of my own first. So one thing about your talk that stood out to me was that these methylation predictors seem to work well across different ethnic groups as compared to other predictors. So I was just wondering if you have any idea why that's the case.
I guess it's probably closer to whether if individuals of any ancestry smoke a cigarette or drink alcohol, I guess it's maybe suggesting the body is processing that in the same way across different groups, whereas with the genetics, it's further from that's a fixed variable, if you like. So the methylation is likely to be changing in response to the environmental stimulus. So perhaps it's tracking that a bit better. It would be interesting to see if we observe similar things for proteins, lipids, and other biomarkers, if they do better than say the genetic predisposition to those the polygenic scores.
Okay, thank you so much. So I will. There's a couple of questions from the audience. So I'll start off with the first one. Do you also observe a regression to the median in older individuals, like how it happens in other clocks?
So for the one we prepared there, oh goodness, can I remember? I think we did see, the performance, it was less well calibrated at the upper end. It was doing better than, or when we fitted the quadratic effects, the quadratic estimates of the CPGs, that did improve the fit at the upper end and the lower end as well. So it's, but interestingly, training on log age didn't make much of a difference at the upper end. So there's also possibly a lack of data in the training set in the oldest olds. So again, if you've got, 5% of the data in the training set come from the oldest people, the model is not going to be performing as well in that age in the test set.
And also another question from Katharina. How much does a recent infection influence the clock?
Oh, I don't know. I think that's one that we would, I don't even know how that would, it's a really good question, actually, or even, I don't think I've seen studies on if somebody, the immediate effects of alcohol, smoking, or whatever the intervention might be, how does that result in changes to the methylome, or is it more of a longer term, or it needs a sustained intervention to have an effect in the mid to long term? And I think that's, my guess is that we would probably see more, it would need to be a fairly long-term exposure before there's reliable readouts. Whereas for things like the proteome, you'd probably see, we know for acute phase proteins, levels can increase, say, 1,000-fold with an acute infection. But the methylome, I think, is more likely to be a bit more stable.
I'm just jumping in. I don't know if this is published, but I know Vadim Gladyshev has looked at that in some of his methylation aging clocks, and they actually see a lot of flux in those clocks. So when you get COVID, your biologic age goes up for a period of time, and then over a month or two, it comes back, hopefully back to where it was before, depending on whether you get long COVID symptoms or not. So that's a different clock. So I don't know how it would apply in yours, but I know that he reported that data.
Interesting.
For the next question, is there any approach or methods that is going to improve dietary reporting based on your experience?
That's an excellent question. It's not something we've looked at. I think that's another variable that's incredibly difficult to measure well, or through kind of questionnaires anyway. It's something that, yeah, we've not explored. I'm not aware of anything in the literature that would be methylation proxies for, say, fruit and veg intake, that sort of thing. Very interesting question, and I would hope that, as with alcohol and smoking, that maybe not methylation, but say proteomics or lipidomics would actually give a good readout of, or a more objective readout potentially of dietary impact compared to self-report questionnaires.
Okay, so for the next question, would you have a more accurate prediction of the biological age of an individual based on methylation when you know the origin of the individual?
I guess ethnicity, or maybe geography. I guess either one of them could apply.
Yeah. Yeah, another good question. I've not seen, I think across the literature that as with the predictors of smoking and alcohol, that the clocks do translate quite well across ancestries. So again, as with most things in kind of epidemiological cohorts and biobanks, the majority of the data at the moment are for European ancestry individuals. So it'd be really interesting to see how well these things translate as we get bigger cohorts, like in like you have in Singapore and across the States as well. That once we get into tens of thousands of multi-ancestry participants, do you get better predictions by just including everyone, or training things within ancestry or within geographic regions?
I think for just jumping in there again, sorry. I think for the SG10K, we have a little bit of insight on that because we have Chinese, Malay, and Indian. And the 4 clocks we used were basically European based on European data. They all worked in all 3 ethnicities in Singapore, but the error rate was much higher. So in other words, you could tell that they were not optimized to these ethnicities. So I think as we get more data, so that means they have meaning looking at other ethnicities, but as we get more data, I think it may be relevant to build ethnic specific clocks and possibly even geographical clocks too at some point.
I think you could maybe do a kind of pan-nation clock if you aggregate existing data sets and find out, say, which CPGs associate with age across all cohorts independently, and then take that subset to train. That would be kind of a fairly simple way. And there will be, I mean, goodness, we see it with the pan-mammalian clock that there are sites that work across all mammals. So I'm sure there's going to be the same case.
We also have another question asking what is the relationship between CPG methylation and type 2 diabetes, and if there is a linear relationship there.
Okay, another excellent question. So we looked at, I guess, combinations of CPGs rather than, so it's an aggregate score. From memory, I think it worked well across, whether we were looking at 2-year, 5-year, or 10-year predictions. So the performance stayed fairly stable over that that period. Again, we explored calibration in the paper as well to see if the predictor works better at depends on the kind of time to event. But we've not specifically looked at individual CPGs and kind of diabetes outcomes and kind of exploring nonlinear patterns there. I think, well, I guess, actually, no, sorry, we did. And so in the previous paper of Yipeng that was published in Nature Aging, we consider random forest approaches and regression tree models to asked, do they do better than elastic net and other methods? But then we were probably a little bit unfair to random forest because we pre-selected features based on linear associations. So you kind of, yeah.
Okay. So since CPGs and other epigenetic changes could vary from one to another, have you considered data integration with samples of different tissues from the same patient?
We'd love to. I guess it's the case of it. It's what's feasible within cohort studies. So we're working with here primarily secondary data analysis. So there's blood, urine, and on a handful of saliva collected 15 years ago. So those were the data that were analysed for Generation Scotland. Moving forward, I think the likes of blood spots, saliva, and kind of just venous blood draws are the most likely or the least invasive ways to capture biosamples. And therefore, they're scalable methods. I think if you wanted to do biopsies of other tissues, it would be A, very expensive, and B, quite invasive. So I don't know if you get the same numbers. It would certainly be interesting to compare across different tissues from the same individual to see, I suppose, as with Steve's original clock, do different organs age at the same rate or different rates, and to do so, and do that within individuals.
Shiva, I'm going to have to jump in here because we're running out of time. With 1 minute left in the hour. Thanks a lot for doing that. And Riccardo, thanks a lot for jumping on the show. It was a great discussion. We'll have to do it again sometime.
Thank you so much.
I want to remind everyone to use the chat function and choose the panelists and all attendees option to leave comments and feedback so everyone can view it. I also want to send out a big thank you to all the participants in our Healthy Longevity Supplements Conference last week, where there were 790 participants from 45 countries and 29 sponsors who were part of the conference. And then afterwards, there was an open day for general public, and we have 520 people show up for that. And finally, we had an intensive course on healthy longevity at the Longevity Academy, and that was highly oversubscribed as well. So I think it reflects the fact that there's so much interest in longevity right now and supplements, and hopefully we can continue that momentum and get the word out to more and more people. If you want to assist the Healthy Longevity Translational Research Program in the areas of research collaborations, media engagement, outreach, and website content management, let us know. You can apply now for a role as an assistant manager. The final video is a 102-year-old fashion icon, Iris Apfel. Actually, I read that she died about 4 days ago. And I don't know why I know that. I'm the farthest thing from a fashion expert that you can imagine, but she did. But this is her sage wisdom for how she got to 102.
Fashion icon Iris Apfel was known for her bold, modern style.
Go wild and get comfortable outside of your comfort zone.
And candid beliefs.
Being stylish and being fashionable are two entirely different things. You can easily buy your way into being fashionable. Style, I think, is in your DNA.
Her signature ensembles, striking colors and patterns, adorned with oversized costume jewelry and her ever-present round glasses.
You have to love what you're doing with your time on this planet, and that's what I've been doing.
The New York native began her career as an interior designer and textile expert, managing restoration projects at the White House for 9 presidents, from Truman to Clinton. Calling herself a geriatric starlet, she was in her 80s when she reached celebrity status in the fashion world.
Get old, but don't get boring.
The subject of a documentary by Albert Maysles called Iris.
I didn't give a damn about going to the party or being at the party. It was getting dressed for the party.
Popular on social media too, with 3 million followers on Instagram. At 97, she signed as a fashion model with a global modeling agency.
It's been a great ride, and I've enjoyed it immensely.
And on her 100th birthday, she had this message on aging and the key to a long and fabulous life.
A sense of humor is absolutely necessary. I don't mean a ha-ha sense of humor, but I mean being able to look at all the silly little things and how foolish they are, and how many important things we really don't pay enough attention to. Be an individual and not to follow the crowd.
Iris Apfel was 102 years old.
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.